All right, so hello everybody. I'm Charles Reese. Feels kind of silly to give this formal introduction since I know so many of you, but I'm a linguist and I'm the director of the Concordia Center for Cognitive Science and Linguistics at Concordia in Montreal. So this year we're pleased to organize this event with the cooperation of various entities at Concordia. We've got the Center for Engineering and Society. We've got the Perform Center, the Linguistics Student Association, and brand new at Concordia is the Applied AI Institute. So I've listed the links for those, all those centers in the chat if you want to check them out. And you can look at those. Okay, so what I'm going to do is we're going to have about a 5-minute presentation or so from Rim Dabbous, who's a master's student, and she's interested in unifying theoretical linguistics and the neurobiology of language using ideas from math and logic and theoretical computer science. And Rim's going to tell us a little bit about Ada Lovelace, and then I'll come back to introduce our guest of honor. Okay, so Rim, I guess you need to share your screen. So I think you can do that, right?
Yeah. You should be able to see my slides. Can you see them?
Yes.
Perfect. So thank you, Charles, and thank you to the Concordia Center for Cognitive Science for organizing this. It's a privilege to be able to talk about Ada Lovelace and her important role in the history of computation. Ada Lovelace is mostly known as the first computer programmer and is famous for being the author of the first algorithm, which is a rigorous description of a sequence of steps to be implemented on her friend Charles Babbage's mechanical machine, the Analytical engine. The algorithm, it's seen here as a diagram for the computation by the engine of the numbers of Bernoulli, appears in Lovelace's notes on her translation from a description of the engine by Italian mathematician Luigi Menabrea. Lovelace's notes and translation were published with only her initials, presumably to hide her gender. Her notes showcase her ingenuity and foresight and are full of insights that transcend mathematics and computer science and align with modern thinking on the nature of computation, artificial intelligence, and cognitive neuroscience. In fact, her speculations were so prescient that she aptly deserves the title of the first computational neuroscientist. As a woman born in 1815, even Lovelace's aristocratic status did not give her access to a school education. Her mother, however, enlisted a string of private math and science tutors to counteract the poetic humors of her estranged father, the poet Byron. And this is an excerpt from a very good graphic novel by Sidney Padua called The Thrilling Adventures of Lovelace and Babbage. So Babbage's analytical engine was never completed. Lovelace's notes and program were based on a mere description of the machine. The engine was planned as a general-purpose mechanical calculator, taking inspiration from the punch card mechanism employed in Jacquard looms. The punch cards, seen here on the left, represented the different programs to weave different textile patterns using the same machine. This card mechanism would have allowed the analytical engine to be programmable by determining the operations to be effected for a given calculation. So from a fixed set of physically encoded operations, the punch card selected a particular subset grouped in a particular manner to achieve a specific goal. Menabrea's original article did contain very basic programs for simple calculations. However, the consideration Lovelace gave to the grouping, repetition, and composition of operations in her own algorithm showcased the first use of fundamental programming principles such as branching and looping. She demonstrates how complex computations can emerge from basic physical mechanisms. Thus, it was Lovelace who saw the full potential of Babbage's ambitious project. She appreciated the analytical engine as embodying the science of operations and saw the significance in the separation of operation from objects operated on, which marks the fundamental transition from calculation to computation. In her notes, she writes, The science of operation is a science of itself and has its own abstract truth and value, just as logic has its own particular truth and value independently of the subjects to which we apply its reasoning and processes. And the bounds of arithmetic were, however, outstepped the moment the idea of applying the cards had occurred, and the analytical engine does not occupy common ground with mere calculating machines. It holds a position wholly its own. Her insights that a machine could compute over any type of symbol, not only numbers, resulted in Ada's famous quote conjecturing that the analytical engine could one day compose elaborate musical pieces. She writes, supposing, for instance, that the fundamental relations of pitch sounds in the science of harmony and musical composition were susceptible of such expressions and adaptations, the engine might compose elaborate and scientific pieces of music of any degree of complexity or extent. In other words, she understood that we can abstract over patterns of textiles, patterns of numbers, and patterns of musical notes, noting that the analytical engine weaves algebraical patterns, just as the jacquard loom weaves flowers and leaves. One of her keenest intuitions relates to the mind-body problem that cognitive neuroscientists are still grappling with. She writes, an enabling mechanism to combine together general symbols in succession of unlimited variety and extent, a united link is established between the operations of matter and the abstract mental processes of the most abstract branch of mathematical science. Thus, not only the mental and the material, but the theoretical and the practical and the mathematical world are brought into more intimate and effective connection with each other. It's unsurprising, in light of the complexity and depth of her conceptions, that she wanted to continue her work by developing a calculus of the nervous system, as she called it. She hoped to create a mathematical model of how the brain gives rise to thoughts. Sadly, Ada Lovelace passed away in 1852 at the age of 36. 169 years later, we look forward to getting from Dr. Hartmann the latest updates on the calculus of the nervous system.
Thank you very much, Rim. That was very nice. Thanks. Okay, I'm going to share my screen for a moment. Okay, let's see. Okay, so Professor Mitra Hartmann came to our first Ada Lovelace talk about a year ago given by David Barner, a psychologist. And I was thrilled when she agreed to deliver this year's talk. We thought that she would get to visit us in Montreal, but obviously that hasn't worked out. I have to say, I've never been very excited about some emerging technology like self-driving cars, but Mitra's project, the digital rat, really caught my fancy. I have to admit being jealous of somebody who has a website that lists her research projects as the mechanics of whiskers, whiskers and the sense of touch, and whisker-based robots. So this gets me excited. So Mitra's training for all this whisker work includes a BS in applied and engineering physics from Cornell, a PhD in, I guess you say, integrative neuroscience or integrative neuroscience from Caltech, and postdocs in computational neurobiology and another one in bio-inspired technology. Mitra is a professor of biomedical engineering and professor of mechanical engineering at Northwestern University. And I just can't resist sharing with you some graffiti that I found about a week ago, biking home. Can you all see this on my screen? Yeah, so there's a construction site. It's an old pool that's being turned into a theater, I think. And I was standing on the pedals of my bike, holding my phone, trying not to fall or get hit by a car. And somebody had written Ada Lovelace on the wall there for some reason. Maybe it was one of my students, I don't know. But you know, I guess it seems that Ada Lovelace too, not just digital rats, are a hot topic, right? The kids who are out doing graffiti are into Ada Lovelace. So we're lucky to have Mitra here to bring it all together for us. So let's welcome Mitra Hartmann. Thank you. Okay, so I'm going to stop sharing.
Sharing screens. Okay.
So Mitra, you ready? You got to share?
Yep, I can share. All right, let me go into presentation mode here. So thank you so much for the invitation to speak tonight. Rim, for that wonderful introduction, to Ada Lovelace, and Charles for the generous introduction as well. So tonight I'm going to talk about rats, and I'm going to talk about robots, but mostly I'm going to talk about closing the sensorimotor loop. So as Rim told us, Ada Lovelace is known for having written notes on the analytical engine of Charles Babbage. And what she's particularly known for is that she had the insight that, in distinction to the difference engine, which is shown up above on the right, which could perform only arithmetic calculations, the analytical engine could combine together general symbols in successions of unlimited variety and extent. And interestingly, I'm going to use the same quote as Rim did, which is that a uniting link is established between the operations of matter and the abstract mental processes. And we might think of these as mechanics, or body, and brain. And so that will be the topic of the talk tonight. Now, I've prepared this talk for a very general audience. So there should be lots of fun videos, I hope, and a little bit of mathematics and equations. But I'm happy to go into more detail with anybody after the talk about any more of the mathematical equations that are relevant here, or any more neurophysiological detail. So again, a very broad talk for a general audience. All right, so I'd like you to think about what happens when you place your fingers on a mug. Your fingers are going to exert forces on the mug, and you're going to experience those reaction forces in or on your fingertips. And you're completely unaware of the space between your fingers. And in fact, you don't even perceive those forces necessarily as forces on your fingers. Instead, you just perceive the mug underneath, right? Those properties of the mug, like it's cylindrical and it might have a smooth texture. Now you might decide, well, you want to change your grip on the mug. Maybe you want to move your fingers along the surface of the mug and grasp the handle. And somebody's taken a photo now from the other side, or you've switched hands. And in order to guide your fingers along that mug, the sensory data that you just acquired from that mug to guide your fingertips along the cylinder just guided your movement. And your movement just changed the sensory data that you received. And together, that sensing and movement is refining your tactile perception of the mug. So you should not think about this sort of thing while you're driving. You don't want to be thinking about how you're perceiving the steering wheel underneath your hands as you steer. It's very distracting. But this is how tactile perception, the sort of loops that occur during tactile perception as we explore the world around us. So what this implies then is that your brain had to evolve to interpret the data from your hands and fingers. And the shape of your muscles and your hands evolved so that the brain could control their movements. And your hand movements evolved to allow your brain to interpret the sensory data from your hands. And so what we're looking at here is, in order to understand the nervous system, the way that the brain evolved, we need to be looking at the loop between sensory data in, how that enters the brain, how the brain controls motion, how you interact with the environment, which then again changes that sensory data. So it's a big loop between sensor, brain, motor, and environment. So this is why you can't just take a cat brain and put it on a rabbit, or a rabbit brain and put it on a cat, no matter how similar they look. The cat's brain has evolved as a predator. Its eyes are facing forward. There's a large binocular overlap. The rabbit's eyes are on its side. It has large ears to sense predators around it. So the brains have evolved for these different sensory organs. So if this loop is so important to understanding the brain, how are we going to begin to study this loop? So my model system of choice is the rat, and this video here shows a rat exploring its world. This is taken in our laboratory. It's exploring some Tinker toys, and what I want you to notice is that it's tapping its nose rapidly on the different objects. It's going at about 8 hertz. And the question might be, well, why is it doing this? Why is it exploring with its snout? So I need to tell you that rat vision is not very good. So over on the left here, we have a picture of what we might see with our human vision. And rats have excellent motion detection. If you've ever tried to pick one up, you know that they can scurry away rapidly, but very poor visual acuity. So they're not good readers, and very poor color vision, right? So they have blurry vision with little color. So how do they explore their world? They explore their world with their whiskers. So the reason that the rat was putting its snout down over things is so its whiskers could touch things. You just saw those whiskers touch that Tinker Toy wheel, and it's going to put its paw right up there, right where its whiskers have been. So this has been slowed down quite a bit. So rats are constantly touching different objects with their whiskers. And those whiskers are going back and forth about 8 times a second. Now the word tapping with a question mark after here is present for me to remind me to ask you to notice whether you perceive those whiskers as tapping on objects. You might imagine that it's just very, very, especially since it's doing it at 8 hertz, very, very fast, it's just sort of tapping against the object. And we'll see later on a little bit in this talk that that's not quite true, right? So it might have looked as though the whiskers are just coming out sort of all willy-nilly from the rat's face, but in fact, they are very regularly arranged on the rat's cheek. And in fact, they are so regularly arranged that we can give the whiskers names. Now, they're not very interesting names, but they're names nonetheless. With the rows, we give letters, so A, B, C, D, E. So the rows of whiskers are given letters, and the columns are given numbers, 1 through 6. And then we have this special column of whiskers in back here. You can see these whiskers that are between the rows, and those are given Greek letters, alpha through delta. And the whiskers are smaller as you move towards the front of the animal, and they're larger as you move towards the back of the animal. And there are no sensors along the length of a whisker. A whisker is like a hair. So you can cut your hair and you don't get hurt. And similarly, you can cut the rat's whiskers and it doesn't hurt the rat. All of the sensing is done in the follicle, just like the hairs on your head. This is going to become pretty important later on. So how are these whiskers moving? You saw those whiskers could move independent of the head. So the way that they're actuated is the whiskers come down through the skin. So the whiskers I've drawn here in green and the skin is in pink. And the whiskers go into these follicles, like the follicles for the hairs on your head. They're shaped sort of like corn dogs in this picture. And then there are muscles that wrap around the follicles. These are called sling muscles because they're wrapped around like slings. And when those slings pull backwards, the skin is going to serve as a fulcrum, and those whiskers are going to protract forward. So I have a whisker here. This is a plastic artificial whisker that we've created. And if you can see my video, I know I'm small, but if you see my video, here's the whisker descending through the skin. I'm going to pull back and the whisker is just going to protract forward like that. So that's how they are actuated. For physiologists or anatomists in the audience, there are also numerous extrinsic muscles involved in whisking, and there are very few proprioceptors in any of the whisker muscles. So there are no sensors along the whisker length. There are only sensors within the follicle, and the follicle has a large blood sinus, which is going to mediate the boundary conditions at the base of the whisker. And the follicle is also packed with mechanoreceptors of all these different types. And interestingly, these are very similar to the types of mechanoreceptors that are found in your hand. Now, I can simplify things a little bit here. What you need to know for the remainder of this talk is that this blood sinus down here is going to change how stiffly the whisker is held at its base. So that's what the blood sinus does down here. And you just need to know that this follicle is packed with lots and lots of sensors. Their names do not particularly matter. So why don't we know yet how the brain works? Right. So for many years, the brain has been studied at various levels of detail: circuits, neurons, synapses, molecule. And every day, thousands of labs record data from all of these levels in dozens of species. And yet, despite this wealth of information, we still don't understand how some of the simplest brain circuits work. So why? Well, a typical neurophysiological experiment, you might present an artificial stimulus, something like this for the visual system, and you give that to the anesthetized or head fixed or constrained animal. And then you record from particular brain regions, either electrically with an electrode or maybe imaging. And then you record a very small subset of neural responses. And so I hope that what you're starting to intuit here is that the the brain does not exist independent of the animal's body or environment. And so this is a very impoverished way to study neural responses. The brain is in continuous interaction with its environment, and we can really only understand the nervous system in the context of the body and environment. And so why don't we do that? Why haven't we done this? Well, the problem is that experiments with freely behaving animals in naturalistic environments are very difficult, and sensory input cannot be measured sometimes in the real animal. So if we had this rat running around, how would we ever compute the signals at the whisker base? How would we compute those signals in the follicle? So we need models to simulate the natural sensory input and the motor output of the nervous system. And so the overriding goal of my lab is to construct a model of the rat whisker system to simulate this complete sensory motor integration loop. So the outline for the rest of my talk tonight is as follows. I'm going to start by telling you some definitions of sensory signals at the base of the whisker. And this will be the most challenging part of the talk right at the start. I need to give you some mechanical definitions and things to remember to answer the rest of the questions. And then throughout the rest of the talk, we'll look at 5 different questions and possibly a 6th if we have time for it. So the first question that I'm going to answer is, how do rats move their whiskers against objects? Are they actually tapping them? And what are some advantages of this type of motion? Then we're going to look at how neurons encode the sensory information. And how can we simulate the rat in its natural environment to try and complete that environmental interaction? And can we build a rat, right? If we understand something, we should be able to build it. And finally, the last question, well, what else are whiskers good for? Is it really just for this tactile sensing? So we'll start with the definitions. This is the toughest part of the talk. So here we have a picture of a rat and a single whisker going down into the follicle. And I'd like you to imagine that there's a force that's been exerted on this whisker. And that force, remember, there are no sensors along this length. That force gets transmitted down to the base of the whisker. And so we can draw in 3 components of force. These are 3 orthogonal components of force. They're perpendicular to each other. And then we also need to draw in 3 moments, okay? What is a moment? Well, you might probably think a moment in time, like an instant in time, but that's not what we mean by moment. What a mechanical engineer means by moment is just a torque. Torque is something that causes a rotation. So a teeter-totter is a best example of a torque, or a lever is a torque. So when I use the word moment, what that means, it means identical to torque. And so there are going to be 3 forces that are trying to translate the whisker at its base, and 3 torques or moments that are trying to rotate the whisker at its base. And so those are all 6 mechanical signals are found at the whisker base, and that's it. That's all the information that the rat has coming in from its whiskers. That is a complete physical description of all the information the rat can possibly get. Okay, so then those signals from this mechanical signals gets transmitted to the mechanoreceptors, all those sensors in the follicle, and the signals go up this sensory nerve. And then there's also a motor nerve that's coming down from the brain, and that's going to move this follicle. So it's going to move the follicle and the whisker. The whole thing is going to rotate forward, like we talked about, like that. And then if the whisker hits something, then this force is going to be exerted. And then just for good measure, we're going to add in the brain, right? So already you see, but even before we added in the brain, it was already pretty complicated. The signals from the sensory side are going to go up through a structure called the trigeminal ganglion, and then into the brainstem, then back out through the facial motor nucleus and down through the motor system. And the rest of the brain is up there, right? For the time being, we just call it the rest of the brain. So a couple of points now, we're going to simplify a little bit. So I told you there are 6 components of mechanical signals down here at the base of the whisker. And I'm going to simplify this for you because it's going to turn out there are only going to be 3 that you need to know. So I deleted 3 of them. We just are left with 2 of the bending moments and 1 of the forces. And I'm going to combine these 2 moments, and we're going to talk about bending magnitude. That is how much the whisker bends. It's going to be how much the whisker bends. The second one is going to be the bending direction. It's going to be the direction in which the whisker bends. And this very last one is going to be the axial force. And that's going to be the force pushing and pulling the whisker out of the follicle. And these are the three variables that I'm going to show you are important for the rat's sensory perception of its world. So again, MB, how much it bends, MD, the direction of bending, and FX, the force pulling it in and out of the follicle. And they're all found at the whisker base here. It occurs to me to ask, Charles, can you see my mouse as I move my mouse around?
Do it again.
You're moving the mouse around. You can. Very good. Thank you. All right. So I'm going to simplify this diagram in another way. I'm going to take out all the rest of the brain. And I want you to notice that the simplest possible loop is just going from the whisker to the trigeminal ganglion, brainstem, facial nucleus, and back out again. And that's the simplest possible loop we can model in the whisker system. So that's what we're going to aim for. All right, so that was the tough part of the talk. So the next part, you get to see some good videos of rats whisking. How do rats move their whiskers against objects? Well, in order to determine this, my student Blythe Towell took a glass sheet shown here in gray. This is just a glass sheet that's about 1 foot by 1 foot. And she took a laser and she turned it into a sheet of laser light that just cascades down in front of that glass pane. Now, when the whiskers break that sheet of laser light, they're going to create a dot of light that can be visualized with a camera here. And there's also a camera up above watching the video taking video from above. The videos are going very fast. We're taking video at 1,000 frames per second. So what you can imagine here is if you have a laser pointer and you shine that laser pointer through the air, you can't see that laser until your finger intersects it and suddenly a dot appears on your finger, right? So that's exactly what's happening here. And we're going to put naive rats on this perch here. And by naive, I mean they've never been on this perch before. They have no idea what's out there in the world. And then we're going to look at how they move their heads and whiskers around. And we're going to look at the first 1 to 2 seconds of exploration. And we want to see how they interact with this glass pane. And so the type of video that you'll see here, you're going to see these little dots of light reflecting the whiskers, indicating the whiskers that are reflecting the light. And there's going to be a nose. The nose of the rat is going to appear in the middle. So here's a video. These are 2 different views of the rat. The view on the left is the top-down view, and the view on the right is the front-on camera view. So I hope you can see the whiskers all breaking the laser sheet of light. In both views here, the nose is very bright because, of course, the nose is also breaking the light again. I'll just play that again to give you another impression of what it looks like in the 2 views. And the next step that Blythe did was to track out each of those individual whiskers. And she created this video that we call painting the whiskers. So it's because we've generated this rainbow color of whiskers. And I'll play this a number of times. This is the pattern that the whiskers are making on the glass sheet. You can see that the pattern is a result of both the whiskers moving and also of the rat's head movements, right? So up here in this region of the video, you see the rat's maybe keeping its head a little bit still. So you can see all the almost the full whisker array here, but then it gradually moves its head around and you can see that it becomes more diffuse. Now there's one other feature I want you to notice. It's most visible over here. Do you see these little V's? V here, a V here, V here. V's are maybe a loop, a loop, a loop, V. The whiskers are sort of being dragged along on the plate. What this means physically is that the whiskers are pushing into the plate and coming off the plate, pushing into the plate and coming off the plate. And remember that a while back I said, are these whiskers actually tapping against the object? They're not. They're pressing in and pulling off and pressing in and pulling off. So I'll play this video again and you can watch those V's occur. They kind of get covered up, but you can see them in orange, blue. And we saw these V's, this pressing in and pulling off, pressing in for every single rat on every single whisk that we tested. It was just a uniform thing that occurred, these types of V's. And we wondered why, what's the point of them? And we noticed that these V's are lasting for a characteristic time, somewhere between 20 and 50 milliseconds, right? So it's for a fraction of this whisking cycle. And so the question then becomes, well, what's special about 30 to 50 milliseconds? Why is the rat leaving its whiskers in contact with the surface for this duration? So we constructed a model of the whisker. Brian, Lucy, Vlad, and another professor at Northwestern, Todd Murphy, constructed this model of a whisker. And what they did is they simulated the whisker rotating at 3 different speeds and colliding with an object. So they're going to take this whisker and they're going to collide it against a peg. So on the x-axis here, we have time. And on the y-axis here, we have the position of the base of the whisker in degrees. So it's rotating. And we're going to look at this, the speed. Let's just look at the fastest speed here, which is shown in dark black. That's going at 1,000 degrees per second. All right, so it's going pretty fast. And we're going to look at what happens when it collides with the peg. And so here's the answer. Well, there's a little bit of startup transient, right? The whiskers kind of got to get itself going here. And then at time 0, bang, it hits this peg and suddenly it starts to bend, right? So it bends, but then it starts to vibrate. So you can see it hits and it collides and it starts to vibrate like this. And all of those vibrations are damped by about 30 or 40 milliseconds. And that happens regardless of how fast the whisker is going. So there are startup transients before time 0, but the effect of the collisions are all gone by about 30 milliseconds. And so we started to think, okay, maybe the reason that the rat is leaving its whiskers in contact with the surface for 30 milliseconds or so is so that these vibrations damp out. And so that leads to the next part of the talk. What are some advantages of this type of motion? So to explore this, we noted that damping, as we hypothesized, or we noted that this damping is going to ensure that the whisker enters what we call a quasi-static regime. And all that that means is that the oscillations are damped out and the whisker's mechanical behavior is going to be described only by bending. And we thought that this would give us a unique relationship between mechanical signals at the whisker base and the 3-dimensional contact point. So let me try to explain this a little more. Remember that I told you that at the base of a whisker, when there's a force exerted, when the whisker deflects against a peg, the 3 signals that we think are important are going to be how much it bends, the direction of bending, and the force that's pushing the whisker in and out of the follicle. But the rat could care less about this, right? The rat saying, where is that point? Like, I just want to know where that point is in the world relative to the base of my whisker. I could care less about these forces. Remember, when you pick up that coffee mug, you're not paying attention to the forces on your hand. You just want to know, is it cylindrical? Is it a smooth surface? You just want to know features of that coffee mug. Where is it? So Lucy Hewitt in my laboratory figured out that there was a mapping between these 3 variables over here, MB, MD, FX, and the spatial position of that point on the whisker on the object relative to the base of the whisker. And the interesting thing that she found, it was these 3 variables, which is why I told you they're important at the beginning of the talk, and it requires that the whisker be tapered. So you cannot have a cylindrical whisker. So one of the reasons we think that rats and other animals have tapered whiskers is so that they can figure out where the whiskers are making contact. All right, so if this is true, if Lucy's work is good, then we should be able to map out, you watch a rat whisk against an object and use these mappings to infer the shape of an object. So let's see if we can do that. So in this video, it's a little complicated, but in the top row here, I've got pictures of a real rat. Okay, so this picture, this one over here, I'll use the annotations. We'll see how we do with that. This picture over here, this is on the top left here. This is the top-down view of a rat whisking against the peg, and the peg appears foreshortened because we're looking straight down on it. And this is the front view. This is the front view of a rat. Here's its eye, and we're looking at the peg from the front right here. And in the bottom 2 images here, this is our simulation of a rat. In the top-down view, you'll see the peg start to appear right here. And this is our simulation of the rat in the front view. There's its ear and nose. So these are, I understand these pictures are kind of hard to interpret unless you have that sort of intuition here. And so what we're going to plot then over here is the shape over here in this picture here. We're going to plot the picture of this peg emerging as determined from these mappings that Lucy created. So top view and front view, we're going to plot the tracked contact point in black. So we went and found ground truth based on tracking the contact point, and the output in pink, that's the output from our simulations using those 3 mechanical variables at the whisker base. And we're going to use whisker position and orientation to put the contacts in lab-centered coordinates. So you should gradually be able to see the shape of this peg emerge. So here we go. So it happens very fast. Remember, the rats are whisking at about 8 Hertz, so this is slowed down. So you can gradually see, so in the top view, the rat is whisking against this peg. And in the front view, it's whisking against the peg. You can see it actually changed, the rat changes the side. The whisker slips past the peg at one point. And here you can see the top-down view of the peg being built up. And here the peg is emerging in front of the rat. And you notice that it almost appears as though there are 2 pegs over here, 2 pegs emerging, but that's actually the front and the back of the peg as the whisker emerges. So it looks like Lucy's mapping is pretty good. I mean, obviously there are errors in it. You know, there's some uncertainty in it. There's going to be a little bit of variability here, and that just tells us that, you know, that nothing is perfect in biology, and that the mappings have a little bit of error in them. There's going to be some uncertainty in it, right? When you go pick up your mug, maybe you slip a little bit, or maybe it's not exactly the shape that you imagine, right? So we definitely expect error in this sort of modeling. Okay, so all right, everything that I've showed you so far is mechanical. What do we expect the brain to be doing, right? And so the question is then, how do neurons encode this sensory information? So remember that this is our loop here. This is the simplest possible loop. The signals are going up the sensory nerve into the trigeminal ganglion, brainstem, facial motor nucleus, and down out. And our hypothesis is that these neurons would encode these particular signals, right? So that's our hypothesis. And so Nick Bush from our laboratory did experiments where he took a rat. This is now going to be a top-down view of the rat. And he inserted an electrode into the rat's brain, and he used 2 video cameras to monitor the position and shape of the whisker as he was deflecting it by hand in 8 different directions at 2 or 3 radial distances and at 2 different speeds. All right, so here's a video of what that looks like. This is the top-down view. And this is slowed way down. So what you're looking at here is the rat, and this is one single whisker coming out of the rat. And this is a carbon fiber rod that he is manually pulling back and forth to deflect this whisker in a particular position. Again, this rod appears foreshortened because it's coming out of the page. Now, this kind of neurophysiological experiment is very unusual. Remember that the standard neurophysiological experiment, you want to give incredibly repeatable stimuli so that you can average, and figure out how does this neuron respond to this average stimulus. Nick is doing this by hand, which of course is not repeatable, right? He's manually stimulating. The idea here is, well, let's cover the variability and stimuli that this rat would perceive and would obtain in the natural world. Like, let's look at what the rat would be getting in natural conditions, not the identical stimulus over and over and over again. Again, speaking to the idea that naturalistic stimuli are important. And so I thought that many of you might want to hear what neurons sound like. And so I hope that you should be able to hear the sound in this next video. If you can't, I will start sharing. I'll restart the share with sound here. So you're going to see this whisker here. This is the 3-dimensional whisker shape, and you should be able to listen to a neuron, the electrical signal from a neuron, as this whisker is deflected. So, and this, this is the shape of the whisker will appear over here. You're going to have 2 views of the whisker in the front and the top view. And here is this bending moment, the magnitude of the bending moment over time. Actually, he's divided it up into all 3 different components. So here we go. Can you hear that?
I didn't hear it.
Okay, let me exit the presentation, and then I will share the sound and reshare the presentation. And then I will start from the current slide. All right, let's try that again. All right, I'm getting a thumbs up. So
Yeah,
you
yeah.
can now hear what that neuron is sounding like. Kind of sounds like a Geiger counter. And what you should notice is that the whisker is responding most strongly when those mechanical signals are high, and particularly when MZ is high. So this particular ganglion neuron was tuned for a direction, a bending direction that had a particularly high value of MZ. Now, not all neurons responded like this. Some of them only responded at the start. They only responded to the derivative of these mechanical signals. No, at the start and end, and at the start. Okay, so those are what neurons sound like, and a representative sampling of the kinds of neurons that we found. And he recorded from 75 different neurons, and he looked at all of the different distances of stimulation out along the whisker length. He looked at the 8 different directions, and he did a number of different speeds, and then he quantified their mechanics in the form of all the moments, the forces, and the rotation at the whisker base. And he looked at the spiking, and he looked at the time derivatives of all of these variables. He was able to split up this population in terms of the very proximal contact, medium contact, and distal contact. So this is how many of each of the types of stimulation he gave. And these are the 8 different directions that he pushed the whiskers. And so the answer is as follows. Remember, our hypothesis was that neurons will encode these 3 particular signals. And he said the finding was yes, but the neurons are not selective, very selective at all. Their responses broadly cover, or they tile, the mechanical space. So the 2 examples that I just showed you was of one neuron that responded very strongly to the bending moment, and another neuron that responded very strongly to the derivative of bending moment. But we found very few neurons on those extremes. I showed you the extremes. Most of them responded to all of the different possible mechanical signals. And so this is very much, this is a new, sort of a new finding. People expected the responses to be tuned to these different variables. It's very much unlike the auditory system, where you have hair cells that are very specifically tuned for particular frequencies. And it has implications for how neurons at later stages of the brain have to process that incoming information. So we are currently in the process of generating models. Remember that I only showed you one whisker, right? He's recording from neurons from one whisker, but there are a whole bunch of whiskers, 31 of them. Here's 5. Each of them has these ganglion neurons associated with it. There are about 200 to 300 ganglion neurons per whisker, and all of those signals get sent to the brainstem. And so we're building models of the ensemble of ganglion responses to send to models of the brainstem. Okay. How can we start to simulate the rat in its natural environment? Well, again, everything we've been talking about so far is just a single whisker. But really, we want to care about the entire whisker array, which looks something like this. And Nadina Zweifel in our lab constructed Whiskit, the physics simulator, where we're going to look at the responses, the mechanical responses of all of the different whiskers in the array. And this is really going to be starting to allow us to look at how the rat might explore the natural world and the kinds of mechanical signals, the input that's coming in during this type of exploration. So I'm going to show you a very complicated video. So I'm going to explain it in pieces. So you're going to see a video. This video will first show a rat with all of its whiskers coming out, and we're going to simulate a peg moving through the array from the right of the screen to the left of the screen. All these whiskers are going to be deflected. And then over here in this part of the video, you'll see a layout of the whiskers down here, each of the whiskers coming out, just like you see the whiskers here. And up here, you'll see cartoons of the schematics of the whiskers follicles. So these are the follicles, representation of the follicles of the base at the base of the whiskers. And you'll see the moments over here in red and the forces over here in blue, and they'll light up when that particular whisker gets stimulated. And the forces will be shown as vectors down in this whisker plot here, and the moments will be shown as red vectors in this plot here. Each of these lines here is a whisker. So we're going to be looking at forces and moments across all of the whiskers. Then down here, we're going to plot the full whisker array, but we're going to flatten it out. So here are all the whiskers from the A row all the way to the E row, from the top of the rat, the dorsal part of the rat, to the bottom of the rat, top to bottom here, the dorsal to ventral arrangement. And we're going to be looking at how those moments are changing as a function of time. And then here, over in this part of the video, moment is a function of time. Over in this part of the video, we'll be looking at time on the x-axis. And again, the whiskers are arranged from the very top row of whiskers to the bottom row of whiskers, from dorsal to ventral, from top to bottom of the rat. And we're going to be looking at where along the whisker length the whisker collided with the peg. So it's a complicated video. There's a lot to see. Here we go. And what I find, so it should be fairly intuitive to you, right? What happens is the peg hits the whiskers and you get signals at the base. Okay, this is what we expect, right? The reason this is fun is that we would never be able to measure this in the real animal. We would never be able to know what the input is. We'd never know how the signals that the rat is getting. And given these mechanical inputs, we can then start to combine that with our knowledge of how these neurons are responding to build this ensemble model of the population of ganglion neurons. Now, this still hasn't gotten, this is a very boring experiment. This is a standard laboratory experiment running a peg through the whiskers. But what about the natural environment? How are we going to deal with that? Well, Nadina went out into the real world, and she took a Kinect scanner and she scanned alleyways in Chicago. And she found a number of places where rats were known to inhabit, and she took 3D scans of back alleyways. So here's concrete ground, here is a drain pipe, here is a wall, and a column is attached to the wall. This is a back alleyway here. And we can start to look at how a rat would be interacting with its natural environment of a Chicago back alleyway. So here we go. We're going to zoom in on that drain pipe, and we're going to watch the rat whisk over the drain pipe. And you can see, the little whiskers here are not actually hitting. They're inside the pipe. It's the larger whiskers out here that are making contact. And so you can see this rhythmic whisking activity and all of the forces and moments that are coming in. So this is the input to the nervous system. This is all the information the brain has to compute the rat's perception of its world based on the whiskers. So this to me is very exciting breakthrough for our laboratory. We've been aiming for this for a number of years. Okay, and the very last question that we're going to look at is, can we build it? Okay, so that was all simulation, but can we build a robot that might perform some of the same functions as this? So Hannah, Matt, Kevin, along with my collaborators Kate Brinson at Duke and Sarah Bergbreiter at CMU, we developed this whisker sensor at the base. It's based on strain gauges. We had to develop a very special way of creating these whiskers. You see that it's tapered. It's very hard to find objects that are tapered in the real world. Like you go to a toy store or a hobby shop, you don't find items that are tapered. We thought maybe broom bristles would do it, but they're not actually tapered. So things are very hard to find. And so the idea is we have this sensor at the base, we can build these artificial whiskers. And so this is a comparison of how our artificial whisker compares with the real whisker. These are not to scale. So this whisker, the real whisker here, is much shorter than the artificial whisker. And notice, interestingly, remember that one of the things we talked about was how important that the whisker is damp. Look at how much more that artificial whisker is oscillating at the end than the the real whiskers. The real whiskers over on the left, the artificial whiskers over on the right, and those oscillations at the end are posing a real problem for us. You can see that in the data here. So we're working on getting rid of those. So Hannah published a paper a few years ago showing that she could use one of these robotic whiskers to perform contour extraction. So you're going to show that this is a cylinder. She moved the robotic whisker against a cylinder and against a cone and was able to pull out the shape of these different objects. And we are currently working on constructing a robotic rat that can go in and tap against different objects. What I say tap, but actually, right, I should say push in and pull out, push in and pull out against different objects and figure out their shape. And we also like our rats to do sort of cute little like goodbyes as they're finishing up, cute little head shape goodbyes. So, all right, so where have we ended up? So we have dealt with the mechanics of the whisker. We talked a lot about that. I put a green check mark by the mechanics of the whisker. And we've dealt with the signals at the base of the whisker, so I put a green check mark there. I have put an image of Yifu up here because something that I did not have a chance to tell you about is our model of how that whisker is going to deform in the follicle and how the muscles are going to make that follicle move. And that's something I didn't have a chance to get into tonight, but Yifu is working on both of those problems. We looked at trigeminal ganglion responses, and that's the work that Nick Bush did, but we did not have a chance to talk about the brainstem, and that was very active work right now. Shanad and I are working on a paper together to describe responses in the brainstem to different stimulations. And so I'd like to conclude by coming back to Ada Lovelace's general comment that there's this link between mechanics and the brain. I hope tonight I've convinced you of that link. And what I think is certainly happening in the brain and in the sensorimotor system is that we have these sequential loops. And I tried to find an image of a spiral, but I really think it's best if I simply annotate it here. I hope you can see what I'm drawing. These sensorimotor loops are what I think is enabling gradually us to build up perception of different objects around us. So that's how I think that we construct impressions of our tactile world. So let me clear that drawing. And I'd like to finish by thanking the organizers of this event. Thank you very much, Charles, Rim, everyone else who was involved, all of my alumni in the current lab, and all of my collaborators, and also, of course, funding sources. If people are interested, I can talk about another function of whiskers that's mostly cool videos. So that could be fun, but otherwise I'll leave it open to questions. Thank you.
Thank you very much, Mitra. That was a fantastic talk. I think we'll spend some time with questions. And I'd like to encourage students to ask questions first. Maybe we could get a couple of questions from students. And remember, Mitra knows that some of you are linguistics students or psychology students or I don't know, whatever else. So don't be shy. This is a talk for a general audience.
Yeah, please don't. I'm happy to, glad for any questions.
Sam, you want to ask a question?
Yeah, I had a question. Can you hear me?
I can't hear you. Your mic doesn't work.
I think what's happening is, do you have the fan on in the background? If the fan is on, it will, Zoom will try to suppress the noise.
Sam's in India.
Is that better?
Okay, good.
Yeah, so I think you said at some point that some of those neurons only respond towards the beginning.
I'm sorry, some of the neurons only respond to?
Towards the beginning of contact, and others respond throughout. So I was wondering, are those deterministic? Like, are there specific neurons that only respond towards the beginning of the sensor, and while others respond throughout, or are these just probabilistic?
Great question. There are these two extremes that I described, one type of neuron responds very constantly to a mechanical input, and the other responds very transiently. Those are two extremes that we find fairly regularly, and those are extremes that many other neuroscientists have observed. And one way to describe them is in terms of adaptation. Like, so the neuron, if a mechanical signal goes high and the neuron only responds once, it means that it's adapted to that signal very rapidly. So we call that a rapidly adapting neuron. And in contrast, if the signal stays high and the neuron keeps firing, maybe it gradually slows down, but it's keeping going constantly. We call that a slowly adapting neuron. So rapidly and slowly adapting neurons are standard in the neurophysiological literature, and we do find those extremes. And in fact, if you use very repeatable, precise stimuli and you average over and over again, you find classes of neurons. You can classify these neurons. But what Nick found is if you use variable stimuli, if you use any sort of naturalistic stimuli at all, these classifications completely disappear. That okay, maybe every so often you hit, very rarely you hit a very rapidly adapting neuron, and every so often you hit a very slowly adapting neuron. But most neurons, 95% of the neurons are in the middle. They're going to be responding with sort of intermediate adaptation properties. And that makes a lot of sense because you don't have precision stimuli in the real world. When the rat's going around the alleyway exploring a drainage pipe, it's not getting perfect stimuli over and over again. It's not repeatable. So does that answer your question?
Yes, thank you.
Maybe Alan, do you want to go? Alan Bell?
Sure, yes. Hi, thanks. It was a great talk. I was wondering, so I've owned a rat and they lose their whiskers sometimes, right? And they grow back. And so I'm wondering about the length of the whiskers and how much that figures into the calculations. And how do the rats know what length their own whiskers are at, if that makes sense, and if that's essential or not?
Yes. So not only do rats lose their whiskers, but if you have multiple rats in the same environment, the dominant rats eat the whiskers of the subordinate rats, right? So it's called barbering, and it's a well-known effect, right? So if you have a weak runt rat, it's not going to have very many whiskers, but if you have a strong rat, it'll have big, strong whiskers. And so they continuously shed and regrow and shed and regrow. When I started a number of years ago, people thought, well, the rat has to have a very precise sense of how long its whiskers are, and that's how they know where it touched something. And part of our mechanical work, part of the reason, one of the things we wanted to show is no, that's completely implausible, because as you say, the lengths are always changing, and it needs to know something about the forces at the base. Now, the interesting thing about the work that I described tonight, where we're looking at the bending of the whisker after the vibrations are damped, the signals that are at the base of the whisker due to bending, they only depend on the length of the whisker from the contact point to the base of the whisker. It doesn't depend on anything past that contact point. So the rat can figure out the contact point based on forces and moments at the base, regardless of how long the whisker is. Now, the vibrations, in contrast, are going to depend a whole lot on how long the whisker is. And you can see that with this artificial whisker that I have here. If I clamp it there, it oscillates one way. If I clamp it much shorter, it's going to oscillate another way. And you can do that with any sort of string or string that's held tight at the two ends. If it's short, it vibrates high. If it's long, it vibrates low. So we think that for the sense of touch where they're bending, that the length of the whisker may not matter a whole lot. It's just trying to ensure that they have a wide range of contact patterns. But for something like vibrations, when they're exploring texture, for example, if the whiskers are vibrating over different textures, or if the whiskers are responding to airflow, vibrating in the wind, then that length of the whiskers will be very important, and the rat will pay a good deal of attention to the vibrations as they're coming in.
So just to follow up with that, so it also made me wonder how much of this could be pre-programmed into the brain, like a whisker DNA versus how much the rat has to get feedback from its environment while it's doing this type of mapping. And I'm sure you must have encountered this problem when you did the modeling of the robots, but how much do you take into feedback in? Just wondering if you could comment about that.
Yeah, so in some ways it's the nature nurture question, and I have no better idea than anyone else. I don't know entirely what's programmed into the rat. I know that the whisking behavior develops very early in young rats, and the rat doesn't seem to have to learn to whisk. It does seem to be an innate motor behavior. The whisking behavior is synchronized with sniffing. So it is very definitely a rhythm that's generated by the animal that it doesn't have to consciously think about, right? So there are lots of motions that we can do with our hands that are completely unconscious. On the other hand, if we're doing a very fine motor task, we might be paying very close attention to our fingertips and the sensory data that we get from our hands. Similarly, the rat, the whiskers are nice. They're very similar to hands in the sense that the rat can move its whiskers back and forth, presumably without thinking about it, via these sort of low-level brainstem reflex loops, but could also, also when it wants to really figure something out, it can exert probably a high degree of cognitive control over the whiskers. So certainly central pattern generators that drive the whiskers, so it's sort of built into their DNA. That pattern is present from nearly from birth, but then cognition and learning sort of come down on top of it.
Okay,
Did
Mohammed.
that answer your question?
Let's go to Mohammed.
Yeah. Hi, thank you so much for the talk. Can you hear me? Yeah, I have a basic question, maybe, and excuse the naivete. It's just so what does this tell us about the brain? Like maybe it's like more specifically the human brain, or how to study it, if there is a connection that we can make to the human brain already, and also like, yeah, basically how do we, or what do you yeah, like how do you expect this experiment, this study to like affect or influence how we study the human brain?
It's a very excellent question. So humans obviously don't have whiskers, right? Or well, we have beards and mustaches, but they're not the same as vibrissal. And so you might wonder, well, how relevant is this to the human system? Something that I didn't really talk about is that there are maps of the body in the brain, right? So the whiskers have this regular layout in rows and columns, and in the rat's brain there are neurons that are grouped in regular rows and columns that reflect that peripheral mapping, the mapping on the face of the rat. And similarly in the human, if you have 5 fingers on your hand, then there are parts of your brain where the neurons that respond to your thumb are adjacent to the neurons that respond to your pointer finger, middle finger, etc. So there are these maps in the brain. So one way that rats and humans are similar is that these maps are present. So other laboratories are studying parts of the brain where those maps exist, and they're learning about the neural properties there. And the properties that they're learning about there in these different cortical regions might easily apply to human cortical circuits. That's one way that they do. From my own laboratory, I think the work that we've done that speaks the most strongly to the human brain is the work from the ganglion, showing that the primary sensory neurons are not responding in these very stereotyped ways that we used to think they would respond. We used to think, okay, they're either going to adapt really quickly or they're going to be slowly adapting. We're going to be able to classify them. But the idea that we really need to use naturalistic stimulation to study how these neurons are responding is, I think, critically important. And I think that result will also be found to hold for the hand, right? So if you go stimulate the hand, the receptors found there are very similar. And I think that people are going to find, okay, if we use any kind of naturalistic stimuli, as you grasp a mug or you grasp something, that we're not going to find these strict classifications of neurons. We'll find something more in between. So I think what I mean to say is my laboratory alone is just can only look at one little tiny part of the brain, right? We can only look at the primary sensory neurons because we do mechanics and the periphery. And I think that our results there translate. Other laboratories are working in more central structures, sort of higher levels of the brain. And I think many of the results there will translate because of these similarities and mappings.
Right. Anybody else? Raphael?
Is there a reason why they have like matrix of whiskers and not simply a column of whiskers? Why do they have to? Why do they have so much whiskers instead of like a single?
Yeah.
I don't know how to say a single column of whiskers.
Why do they have so many of them? Or another way to ask it, so why do they have so many of them? Why don't they just have a single column? Or why don't they have a full koosh ball of whiskers? Why do they bother to move them at all? Why aren't they just completely surrounded by whiskers? Both of those are good questions. So one of the pieces of work that we did was to look at what we call the sensory volume of the rat. So that would be the region of space that the whiskers can extend. And we think that the number of whiskers and the volume that they extend out in front of the rat is related to the locomotor speed of the rat. So the rat tends to put its paws down into the region that it just whisked over. And if you could imagine, if you just had one column of whiskers, there wouldn't be enough of them to sort of cover that space, so that you need enough of them to cover that, to encompass that full volume. So that's why you wouldn't have one column. Another interesting question is why you wouldn't just have whiskers coming out constantly from the face. Why do you need to bother to move them at all? And I think the answer there is, well, you get a lot more information if you move your whiskers rather than just having them passively stimulated.
Richard?
Good evening. It's a real treat to listen to you.
Hi, Richard.
Hi. You talked about space and the representation in space. I'm always wondering about time and the way that whiskers move. Do you think that the primary sensory neurons are tuned in time with the regular movements of the whiskers? Like when they're exploring with their whiskers, is this better for the primary sensory neurons, or when you gave stimuli where you pushed in a passive way, if you will? So what's the difference between the active and the passive?
Oh, great. Yeah, great question. Okay, so do you remember the graph that I showed of the response when the whisker collided against the peg? And I said there was a startup transient, and then it hit the peg, and the signal got a lot bigger and went down. Okay, so that's what that startup transient tells us, and what we can confirm is that even if the whiskers are not hitting anything, not making contact with anything, they're just sort of going back and forth here in free air, free air whisking, there are still signals that are generated in a number of the ganglion neurons. And what those are telling the rat is, where am I in my whisk cycle? Am I down here, or am I up here? If this is a rat whisking back and forth, where am I in that periodic motion? And I think something on the temporal, for temporal information, what's going to be important to the rat is where within that cycle, in what phase of that cycle did I make contact with something? And that timing information will almost certainly be very important towards localization of an object. So I do not mean to imply that the mechanical signals, that space alone is telling us where things are, right? The timing information is certainly important for to solve the localization problem.
I have a question that's kind of related, I think. So, like sometimes psycholinguists use eye tracking experiments or and basically you can see when people are surprised in some way. And I'm just wondering, is it possible to, get a rat kind of bored? You keep showing it, you keep putting it in a place where it feels things of the same shape, and then you give it some really, really weird shaped object. And do you notice that it's actively exploring? I mean, is there a way to see this, to see that it's consciously or exploring the new thing?
Yes, this type of novelty task I know of it in the context of some work on the superior colliculus, which is another brain structure. So what they did is they it's a novelty exposure test, and they just measure the amount of time, the duration that an animal spends exploring an object. I think they do similar work on babies, right? If anyone does baby psychophysics in this group. So you just watch, you present a mouse with a cylinder, say, and it's a new cylinder, and the mouse explores this for some duration, and you bore the mouse with the cylinder, and suddenly you present a cube, and suddenly the mouse goes, oh, the sharp corners, they get very interested. So the duration experiments, measurement of duration works there. You have to be very, very careful to look out for scent, because these animals are so olfactory in a way that humans just aren't. So the big problem with rat psychophysics, or rat behavior as it were, is the odors that we can't smell. They're very sensitive.
All right, students, you shouldn't be shy anymore. Any student questions? Any more student questions? We had a few. Sarah, how about something philosophical? I do this in class. I put them on the spot, so they're used to it. Rim, you have any questions?
Actually, I have a question with regards to the maps. So when you speak about the homunculus, that whisking systems similarly have some sort of cortical map representation. So could you elaborate a bit about that, and whether there's similarly to vision, for example, or the sense of touch, whether there's cortical magnification and what that entails?
So the person who's done the most on cortical magnification that I know in the somatosensory system is Ken Catania at Vanderbilt. So he's done some beautiful work comparing, I think, cortical expansion compared to peripheral surface area. So that's not an area that I know well. There are other people far more expert in it than I am. There are, in the same way that there are cortical columns in visual cortex, there are cortical columns in somatosensory cortex. It's called barrel cortex. It's called barrel cortex. Barrel cortex is the whisker representation in somatosensory cortex. And it's called barrel cortex because it was found when people stained that region of the brain, and the staining revealed patterns that look to them like beer barrels, big barrels of beer. And so they call it barrel cortex. And this is the most famous part of the rat brain, is barrel cortex. And those are the cortical columns. Just like in the visual system, layer 4 is the input, and it has similar very large projections from cortex back down to thalamus. The area of somatosensory cortex that responds to the whiskers is greatly magnified compared with the rest of the rat's body. I hope these are pointing towards answers to your questions. I can't speak to the question of cortical expansion compared to peripheral representation. I don't know the quantitative answer there.
That answers it. Thank you very much. Actually, if I may, I do have one other question. It doesn't really, I'm wondering, I don't have that much knowledge about this, but I know that there are grid cells in rats to determine the location in space. So do the whisking have any bearing on that, or is it?
I just saw a couple of scientific headlines on that, and it's not a literature that I know, but it's a fascinating topic. And when people study the hippocampus, they're often looking at visual stimuli. And for rats and mice, I think that whiskers and olfaction would be great stimuli to try. I think maybe Eichenbaum has done some work on that.
Okay, we have a question from Georgia.
So I'm just wondering, because there's repeated stimulus on the rats, have you ever encountered whisker fatigue, and does that affect what you see in the representation of sensory stuff? The
only time that I've heard of whisker fatigue is for cats. Is that where you've heard of whisker fatigue?
Yes, I
Yeah.
assumed that it was all animals with whiskers.
I've only heard of whisker fatigue affecting cats when their water supply is placed in bowls with high edges. And so they have to push their whiskers against the bowl in order to drink. And they don't like that, right? Our rats do not have that type of water supply. And I think what's disturbing to them is the large amplitude bending. And for a rat in the normal world, like a free rat running around, it could always modulate how much it pushed its whiskers against things. So we've never forced our rats to push the whiskers through very large amplitudes against objects. So we haven't encountered whisker fatigue because we don't exert those high amplitude displacements. But look out for your cats. Yeah, give them shallow basin water dishes.
Richard, is that another question or is your hand still up?
It's another question. I'm exaggerating, but anyway.
No, go for it.
This is interesting. So if you're building a whisker, and you're building a rat from the mechanical properties, rats right now, from what we know, they're 8 Hertz machines. They sniff at 8 Hertz, they chew at 8 Hertz, they whisk at 8 Hertz. But if you build one and your whiskers are a bit thicker or a bit different, and you've already said that the mechanical properties of the reverberation, the mechanical aspect. So do you think that, you need to build one that oscillates more slowly at the beginning, or some way of matching? Essentially, is the 8 Hertz things that we see in the animal based on the whisker characteristics, the mechanical characteristics? Other people, have a bias that they think it's the way the wiring works, but what about the whisker dictating the frequency of whisking? I'm just wondering about that.
Very good, very good. All right, so lots of sort of deep ideas there. The first one is the rat is an 8 Hertz machine, except they do lick at 6 Hertz. I don't know, who knows why, right? So the licking doesn't seem to be synchronized. It's a little strange. And the ideas to which you refer are called dynamic scaling or dynamic similitude. And so one of the classes that I teach is fluid mechanics, and dynamic similarity is very important in this field. And what you're pointing out is that if you have whiskers of different sizes and masses, that their mechanical behavior will be very different. So let me give an example to give some intuition, and then I'll come back to whiskers. If I'm a tiny little cockroach and I'm running along, I don't care if I run into an object. I don't care if I run into a block. I'm happy just to crash into that object, fling myself around, like tumble over backwards. Like I'll just run right over that object. How I get over the object is I bounce against things. And I can do that because I'm so low mass. And so the collisions don't affect me that much. If I'm a human and I run into a block, I have such a big mass, right? Like that's not the way that humans want to get over obstacles, right? Cockroaches, this is what they do. Insects, this is what they do. And this has to do with the mass and the dynamics of the system. So what we have to do when we're building our robotic whiskers is to compensate for that extra mass so that we ensure that particular quantities are held constant. And in our case, what we're going to hold constant is something related to Young's modulus divided by the density. So Young's modulus has to do with how stiff a whisker is at each of its, how stiff, it's a material. Young's modulus is a material property. So it's intrinsic, it's an intensive property, it's a property of the material, and it tells you how stiff the material is going to be, how much it's going to resist bending. And of course, the stiffness, though, is going to depend not only on Young's modulus, but also on the geometry, right? So as the whisker tapers, see if I have a less curvy whisker, as the whisker tapers, the stiffness, Young's modulus will stay constant, but the stiffness will decrease as you go out along the lengths. And then, so Young's modulus, then, we divide that by the density, and there's some other factors in there as well, but the density, of course, is related to the mass. And so it's a stiffness mass calculation. That's going to tell you, it's going to be related to how much the whisker is going to droop under the effect of gravity. And it's going to be related to how much it resonates and the vibrations. And so we can keep these particular dimensionless parameters constant. So Young's modulus over the density is not dimensionless, but we throw in a couple of other factors and it becomes dimensionless. Those are the important parameters.
Anybody else? Last call. Sarah, I knew you'd had one in you, Sarah.
Thank you very much for the talk. It was really interesting. I have a question. You mentioned throughout the talk the importance of actually testing the rats in the wild. And I was wondering if you noticed differences between rats in labs and labs in the wild, and if you were able to distinguish between a difference in environment versus a difference in the brain. So lab rats having different brains than wild rats, basically.
Oh, that's a beautiful question. Thank you so much, Sarah. So I have not studied wild rats compared to laboratory rats, but there is a professor at Oxford who did, and he made a whole long video on it. And the video is called The Natural History of the Laboratory Rat, and you can find it on YouTube. It's about half an hour long, and what he did is he took a cage of laboratory rats and he let them out into the wild. So the students in my lab like to call it the Rats Gone Wild video. And what he showed is that these laboratory rats very, very quickly adapt the same behaviors as a natural rat, a wild rat would. So they adapt incredibly quickly. They try different foods in the same way. They start making running trails in the same way. And so it's an interesting question then whether their brains are different. Presumably they'd be more different if it was at birth. I don't know the answer to that question, but one of the students in my lab, Chris Brzee, he went on to a postdoc with Leah Krubitzer at UC Davis, and he is currently studying the differences in the brains between of wild raised animals compared to laboratory animals. So people are looking at this question.
One more and we'll stop at 7:30. Alan, is that another question?
Yeah, it's another question, just a quick one.
Yeah, good.
Because we have been talking about rats, and you had a picture of cats and rabbits at the beginning of your slides. And so I'm wondering how much of the information that you're studying is rat specific versus whisker specific. Do cats and rabbits use whiskers in the same way? I know they don't move them in the same way, but do they use them in the same way?
I might actually have a slide on that. But it's going to be hard to pull up, I guess. So cats do not whisk. They don't move their whiskers back and forth in the same way as rats do.
Isn't this in your video that I saw? One of your videos covers that, right? The science, one of the videos.
Yeah, that's right. So cats, they can sort of protract their whiskers a little bit, but they don't move their whiskers back and forth rapidly like rats and mice do. Whiskers have evolved lots of times through evolution, right? So it's hard, you can't really say there's a single line of evolution that causes whiskers. But one question you might reasonably ask is, well, why do animals like deer and tigers and a fox and dogs have whiskers? Because they're certainly not whisking back and forth. Like, what are they doing with whiskers? And that is actually, that's the what else are whiskers good for part of the talk, which is they're good for sensing airflow. And so when airflow, these animals are all really good at sensing which way the wind is blowing. And the reason is that they need to tell the direction of the wind to tell which way an odor is coming from, right? The animals find mates and water and food sources, predator, prey, all of those are carried by odor plumes, which are patchy. And so they have to know, oh, the wind's coming from over there, wind's coming from over here. So one of the things that I think all mammals with whiskers are doing is sensing which way the wind is blowing. So cats are probably not using their whiskers in the same way as rats to do the sort of tactile exploration through whisking. Something that cats do a lot with their use their whiskers a lot for is climbing. So climbing seems to be associated with high whisker use. Rodents climb a lot, cats climb a lot. Dogs don't have such elaborate whisker arrays, and dogs are not very good climbers. So there's an association with climbing, but again, whiskers have evolved so many different times that it's hard to make a particular correlation with anything. We recently did an experiment to show that rats do use their whiskers to figure out which way the air is blowing. Figure out where the wind is blowing.
Great. Okay, I think we can thank Mitra one more time, and that was really
Thank you very much for the
different
invitation.
from what many of us are used to, and yeah, it's very enjoyable. Thank you very much. So Mitra will be in touch, and Mitra's actually been asking me challenging questions about linguistics too. So I'll try to come up with some better answers for you.
All right. Well, looking forward.
Okay. Have a good evening.
Thanks.
And
Thank you.
Thanks everybody. And thanks Rim. That was an excellent overview of
Really good.
Ada Lovelace. Thank you very much.
Thank you everyone for coming.
Okay. Good night.
Thank you.
Good night.
Good night.
Bye-bye.